Cystic fibrosis is a complex disease that affects multiple organs, including the lungs, where it causes persistent infections. These infections occur because abnormally thick and sticky mucus prevents the cilia, small hair-like structures on top of cells lining the airway, from moving dirt and germs out of the lungs. Cystic fibrosis is the result of changes, or mutations, in the CFTR gene, a piece of genetic code found in DNA that tells cells how to make CFTR protein. The CFTR protein helps cells control the movement of salt into and out of the cell and fluid across the airway tissue, which keeps mucus at a healthy level inside the lungs. But with CF, the mutated genes give cells the wrong instructions, so they make an abnormal CFTR protein that does not function, resulting in the buildup of mucus. Fortunately, promising therapies are being developed to treat the genetic cause of the disease. One treatment being developed is RNA therapy. When the CFTR mutation appears in a patient's DNA, it also shows up in the messenger RNA, which is a replica of the DNA sequence. This mRNA carries instructions out of the cell nucleus to ribosomes in the cytosol, where proteins are made. Mutated RNA makes a non-functional protein, too little protein, or no protein at all. If successful, RNA therapy would either replace the faulty messenger RNA with the new mRNA with the correct instructions, or repair the defective RNA. Once inhaled into the lungs and entering airway cells, new messenger RNA would be able to make normal, functional CFTR protein that moves to the surface of the cell, where it adjusts the amount of salt and fluids flowing into the airway space. The restored flow returns mucus levels to a healthy state, making it possible for the cilia to move freely, removing dirt and germs from the airway. Another possible way to restore CFTR protein is gene transfer. Instead of inserting RNA, gene transfer would work by inserting a normal CFTR DNA sequence into the cell so that it can make functional CFTR protein. Gene therapy could be inhaled or given through an IV. However, getting DNA into lungs is challenging. Cells are surrounded by protective membranes that are designed to keep out foreign objects. And in the lungs of people with CF, cells are also covered with a thick layer of mucus. Specialized delivery mechanisms called vectors are being developed which would take the new correct CFTR gene into the cells. There are multiple types of vectors derived from viruses or synthetic molecules. Gene transfer could potentially work on anyone with CF, no matter what kind of mutation they have. Gene editing in cystic fibrosis would repair the CFTR gene mutation in the person's own DNA. There are multiple gene editing techniques including CRISPR-Cas9, zinc finger nucleases, transcription activator-like effector nucleases, and others. Each of these techniques remove the mutations from the gene. To repair the damage, the cell puts in the correct DNA sequence for a functional CFTR gene. Gene editing can be done with cells inside the lung or in cells that have been taken out, corrected in the lab, and put back into the lung. Gene editing can either be tailored for each specific CF mutation or designed to correct most or all mutations. Gene editing changes the DNA permanently. So if CFTR mutations in stem cells can be edited, all daughter cells derived from the edited stem cells will make correct CFTR protein, making repeated treatments unnecessary. We are at an extraordinary time where science has made incredible advances. However, there is still critical work necessary to develop these therapeutics so they will benefit all people with CF and, one day, provide them with a cure.